everybody, Dr. Ahles here. In this short video, we're going to talk a little bit more specifically about the cardiac muscle tissue that makes up the heart, and we'll also compare it to the skeletal muscle tissue that we focused on last semester. When we talk about cardiac muscle tissue, here's a little bit of a review for you of some of the important things about it. The first thing about cardiac muscle tissue is that we call it a striated muscle tissue. A striated muscle tissue is like we see up here, where I have these kind of checked lines that go back and forth on these muscle cells. This is actually a similarity that cardiac muscle has with skeletal muscle. This striated pattern that we see, again, here's some of those stripes up here. This is due to the arrangement of our actin and myosin proteins. So they're arranged into those sarcomeres, just like we see with skeletal muscle tissue. But there are some major differences as we look closer at cardiac muscle tissue compared to skeletal muscle tissue. One of those differences are what we call intercalated discs. So intercalated discs, these dark bands that we see throughout cardiac muscle tissue, they have two different kinds of structures in them that are unique to cardiac muscle. The first structure that we see in these intercalated discs are desmosomes. If you remember from A&P1, desmosomes are what give us the strong attachment between different cells. So we see this both in the skin and again here in our cardiac muscle tissue. So desmosomes anchor one cell to the other. This is really important when you have a type of tissue that's constantly pulling away and toward each other during the process of a heartbeat. So in these dark spaces that we see here, we have lots of desmosomes holding those muscle fibers together very tightly. We also have what are called gap junctions though. And when we think about gap junctions, the goal of these junctions is not to strongly attach cells together, but to electrically attach them together. So what I mean by that is when I have a message inside one cardiac muscle cell, remember that that message is an electrical charge. So if this muscle cell has an electrical charge, it literally has little tunnels to just send that electrical charge to its neighbor. By having these gap junctions, cardiac muscle tissue cells are able to function together, or they're able to contract at the same time because they're all receiving and sharing this message together at the same time. So when we talk about cardiac muscle cells contracting, this was one of our types of muscle tissue that was involuntarily controlled, meaning you don't decide when your heart beats. And that's a good thing, right? We don't want to spend our entire life telling our heart to beat. But when we think about the way that cardiac muscle tissue contracts, we have something that's called a syncytium. I want you to underline, highlight, star that word. You don't have to be able to say it, but we do need to know what it means. So when we talk about cardiac muscle tissue as a syncytium, what we mean is that large groups of individual cardiac muscle cells all contract at the same time. This is because we have those gap junctions where I can send those messages to tell all these cells to contract together at the same time. Now we see two main syncytia, or two of these groups of fibers that contract together. Unsurprisingly, the first group is in the atria, those top chambers, and the second group is in the ventricles, those bottom chambers. So we contract these cells together as groups called syncytium, and we can do that because they're sharing their messages with one another. So be extra familiar not only with what these structures that we see and their functions in cardiac muscle tissue, but make sure for lab that you can identify what this tissue looks like, and especially that we can identify those intercalated discs. I want to provide you with a little more detail in comparing cardiac muscle to skeletal muscle. So when we're comparing them, there's a lot of different ways that we can. I'm going to highlight a few of them that are extra important. So the first comparison we can make is differences in their structure. We already talked about how cardiac muscle is striated, it has stripes, just like we saw with skeletal muscle. But remember from A&P1 that skeletal muscle cells were really long and were kind of like a cylinder. Cardiac muscle cells, in comparison, are really short. And instead of just being one long cylinder, cardiac muscle cells branch into multiple different types. When we talk about connecting cardiac muscle cells to each other, we had those gap junctions, whereas with skeletal muscle tissue, we did not. And this is why skeletal muscle tissue does not have this syncytium effect. So remember, syncytium means that we're able to contract a group of things together at the same time. We don't do that with skeletal muscles. They had these things called motor units, where we contract a small little group of skeletal muscle cells compared to our gap junctions, where we use that to make this syncytium. As we consider contractions of cardiac muscle tissue, I also want to point out a couple of big differences between the contractions. One of them is that cardiac muscle tissue has a longer contraction period, meaning from when I start to contract to when I finish contracting, much longer in cardiac muscle than in skeletal muscle. This is because for me to fully contract and do my purpose, I need a lot of time to be able to get all that blood out of a chamber. So all the muscles in a ventricle will contract, but they're going to contract for a long period of time to make sure everything gets pushed out before we're ready to relax. The other thing about cardiac muscle contractions is that they have a longer refractory period. And remember that a refractory period was a period of time where I can't hear any other contraction messages. I'm trying to restore my chemical balance. I'm not listening for a message that tells me to contract. When we're talking about cardiac muscle tissue, the amount of time that it waits before it can hear a new message, that refractory period, this is exceptionally long compared to skeletal muscle. This is because we need all of our blood to be able to flow back into a chamber. We have that period that we'll talk more about called diastole, where the muscle is relaxing so that blood can flow into a chamber. So, big difference in contraction between skeletal muscle and cardiac muscle. We use these groups of cells contracting together, called the syncytium, to function as one unit. And when that unit contracts, it contracts for a long time, and when that unit rests, it rests for a long time as well. The only other thing I want to focus on right now is this last note about the way that these types of muscle tissue make their ATP. Skeletal muscle tissue can use aerobic respiration, that's when the mitochondria make ATP, or anaerobic respiration, meaning we can do this process called glycolysis to make ATP even if we don't have oxygen. But when we talk about cardiac muscle tissue, they can only do aerobic respiration, meaning that we must have oxygen for a cardiac muscle cell to be able to make any ATP. This is why in our previous video we talked about the importance of the coronary circulation. The coronary circulation makes sure that all of my cardiac muscle cells have the oxygen they need to continue to make ATP. So as you're studying cardiac muscle, study all of the important features of that muscle itself, but also be familiar with how that type of muscle tissue is similar to or different from skeletal muscle.